All right, it is 6 p.m. in Lisbon and London, and we begin our fourth Janeiro-na-Madeira Winter Summer School this year. We have more than 20 people tuning to hear the first lecture, and without further delay, I give the word to Muriel Priot, who will announce the first speaker.
Yes, so ladies and gentlemen, sorry, I was muted. Okay, so I was saying that 4 years ago, when the first JNM meeting was launched, I remember going to Roman and telling him, do you know who was in the audience? Jean-Pierre Changeux was in the audience. And then from 4 years from that time, we now welcome Jean-Pierre to give the opening conference of the 4th edition of the JNM meeting. So Jean-Pierre has been a visionary thinker very early on in his career. He has dedicated his remarkable career to unraveling the complex mysteries of the brain and shaping our current understanding of neurobiology. He identified and purified the nicotinic acetylcholine receptor, but he is also known by generations and generations of students who, like me, sweated and sometimes dreaded the equations describing the famous Monod-Weyman-Changeux model of concertine transition for allosteric proteins. His academic journey began at the prestigious École Normale Supérieure de Paris, where he obtained his bachelor degree. He was then given the extraordinary opportunity to be trained as a PhD student under the supervision of no other than Jacques Monod and François Jacob at the Pasteur Institute, two great figures of science in France as well. Then he left France for California and then New York to finally come back and be appointed at the Pasteur Institute. Beyond science, beyond the lab, Professor Changeux has met the challenge to bridge the gap between biology and philosophy. His exploration of the neural foundations of consciousness and creativity has made him an accomplished author with more than 600 scientific articles and several books. And you may want to check his latest book, in which he was nice enough to share with me, The Beauty and the Splendor of Truth, Interviews on Autobiographical Recollections. Proof that Professor Changeux is a national treasure in France. He was distinguished by countless awards and prizes. He is a member of the French Academy of Science and was, of course, awarded the gold medal of the CNRS. He has also received numerous international awards, including the Albert Einstein World Award of Science conferred by the World Cultural Council. His influence extends far beyond academia. He keeps inspiring generations of researchers and thinkers. It is an honor to welcome Professor Jean-Pierre Changeux to this meeting, and thank you so much, Professor, for accepting our invitation. And now I leave you the floor if you want.
Thank you very much, Professor Muriel Priot and Professor Roman Zubarev for this invitation to give the opening lecture of this Winter Summer School in Madeira. And it's both an honor and a pleasure for me to talk about Allostery, retrospective and new perspective, let's say from bacterial regulatory enzyme to the brain and the mind, if I may say. This general idea and framework is all of us to try to understand life. And we may start with Claude Bernard, milieu intérieur and homeostasis, and there is indeed the ability to maintain a steady internal environment regardless of the external environment. And more recently, Norbert Wiener and cybernetics develop a theory of control and communication, which of course account for homeostasis in animal and the machine. he mentioned the robots. I feedback, therefore I am. And the interesting outcome of cybernetics was the development of the application to biochemistry and metabolism, and the discovery of feedback-regulated enzyme. The first by Umberger on amino acid, and Pardy on nucleotide, which are of course of great importance to build up the DNA. And I will therefore start to discuss what we know about allostery from the genome to the mind, if I may say, but very concretely on first allosteric regulatory enzyme and the definition of allostery, if I may say. The story started with the discovery of Umberger that I already mentioned, that in the pathway of biosynthesis of L-isoleucine in bacteria, there exists indeed a feedback regulation of the first enzyme by the end product of the pathway. And in addition to this feedback inhibition, there are some cooperative effects which Umberger noticed in the response to the feedback inhibitor. And I started my thesis work to try to understand the actual mechanism of this feedback inhibition of an enzyme which substrate is far different from the feedback inhibitor. I saw there was some kind of special mechanism involved at the protein level on this first enzyme. And my first aim at the beginning of my thesis work would try to uncouple the catalytic activity from the feedback control. And indeed, this was achieved by heating the extract of the enzyme and still keeping the activity, but eliminating the response to isoleucine, as well as the cooperative interaction I just mentioned a few minutes ago. This was actually published in the Spring Harbor book and presented in the symposium in 1961. And in this very first paper, I proposed already two kinds of models. One where the isoleucine and threonine interact through some kind of competitive interactions through overlapping sites. And the alternative hypothesis was to postulate that there were two different categories of sites, a site for the regulatory ligand and a site for the catalytic activity binding the substrate of the enzyme. And of course, this topographically distinct interaction would be mediated by a conformational change. So the word I use, no overlapping, was not attractive enough. And in the conclusion of the meeting, Monod and Jacob introduced the word allostery to qualify this model 2, and this has been since a very popular name. Now, parallel results were obtained by another bacterial enzyme, aspartate transcampamylase, and the uncoupling also through Cyril group reagent by Gerhardt and Pardy. So this work and the subsequent work of my thesis on the property of the threonine deaminase and on the cooperative interaction and the modulation of the cooperative interaction by regulatory ligands like isoleucine or valine, led to the proposal of a rather, I would say, original kind of scheme, because we had to deal with a very complex kind of interaction between these different sites. But to deal with it, we propose a very simple-minded model, which is based first on the fact that the cooperative effect I just mentioned were due to the cooperativity of an oligomer made up of several subunits. You can see two here. That this oligomer possess symmetry property and undergo a conformation change between, let's say, a relaxed and a tense state where the quaternary change takes place, quaternary organization of the protein structure. And the interesting original postulate was that these states pre-exist in conformational equilibrium with an equilibrium constant L, and ligand shift the equilibrium and in fact activate or block the enzyme in one of these two conformations. So this was further expressed in the end of my thesis work, and most of all in this paper with Monod and Wyman, where we formalize this kind of scheme. And one of the main aspects of the formalization was not only the binding of the ligand to each conformation and so on, but the distinction between two functions, a state function for the conformation and a binding function for the ligand. And from an operational and experimental point of view, this has, as you shall see, a very interesting prediction that the state function should be distinct from the binding function, which is simply due to the fact that the states pre-exist in conformational equilibrium. So this is what we may call a minimal model for signal transduction by selection of pre-existing conformation of cooperative oligomer. And if you like the philosophical interpretation of it, is some kind analogous to Darwinian selection of spontaneous variation. Now, at the same time, Daniel Kochland, or even before for enzyme activation, proposed the concept of induced fit in the sense that the ligand cause, as you can see here, a change of a conformation when it binds. So there is an induction of the conformation change, and to account for the catalytic activation of the enzyme. So this was not proposed in the case of allosteric interaction, but of course could be extended. That what he did after us, Kochland and in Filmer in 1966, in a model that you can see here, where the conformational change follows the ligand binding or concomitant with the ligand binding without, of course, any kind of pre-existing conformation. So this, to some extent, I may qualify it of a Lamarckian process where the function creates the organ. Now, this model has been wonderfully demonstration by Iwata with Bifidobacterium longum, where he was able to co-crystallize the two allosteric states of this actate dehydrogenase that you can see here. You saw also the symmetry of this beautiful tetramer. And you can also see that the conformation change from R to T does not affect so much the tertiary structure, a little bit, of course, but mostly the quaternary organization through some kind of rotation of a few degrees. And most of all, which is an interesting feature that we shall again see later on, the binding site, the catalytic site, and the regulatory site were located at the boundary between subunits, which was, of course, very convenient for having the conformation change at the level of the quaternary structure, changing concomitantly the catalytic and regulatory site. So this is, if I may say, quite a beautiful static demonstration of the model, and that is consistent with the model. and I went to do my postdoctoral stay with John Gerhardt and Howard Chapman in Berkeley, and then the opportunity to try to compare the state function, remember R against Y, and demonstrate that in fact the R function is different from the state function due, as I said before, to the occurrence of pre-existing site and the shifting of a population from R to, of conformation from R to T, or from T to R. And therefore, we may say that this experiment and others which have been done support, I don't want to say that fully demonstrate, but at least are consistent and support the two-state model compared to the induced fit one I just mentioned before. And therefore, at this stage, which has been continued, of course, with many enzymes and still going on in many laboratories, the challenge between MWC and quotient is still debated, but there are abundant evidence supporting the MWC model. Now, in my thesis work at the very end, I raised the question how general is this model can be extended to brain communication. This was in 1965, quite a while ago. But at that time, of course, the communication in the brain were known to be either electrical for the propagation of the nerve impulse on the axons, or chemical at the level of the synapse, where a neurotransmitter is released, for instance, acetylcholine, in the synaptic cleft, diffuse, and then bind to a receptor, which is present in the postsynaptic membrane. There are abundant neurotransmitters, at least 10 to 20 small molecules, many neuropeptides, and therefore the brain, in fact, is not simply an electrical machine, as it was thought by Eccles and others, but a highly diversified chemical machine where there are transition here from, for instance, close to open channels or enzyme activation taking place at the level of receptors. So at that time, there was not a single receptor being identified, and to follow up on the idea of my thesis proposal, I tried to work in David Nartman's lab and work on the isolation of the nicotinic receptor. And the first experiments were done on the fish electric organ, which was shown to be extremely rich in cholinergic synapse, all of the same kind, and with the properties identical to those of the neuromuscular junction in humans. I was lucky enough to meet Chen Wanli, who had developed snake venom toxin, which appeared to be exquisite labels from the receptor from electric organ, and then to isolate the protein, to purify it, thanks to this toxin. And here is the first image of any receptor seen, ever seen at the time, of the receptor from electrophoresis, the electric organ. And here you can see from torpedo the postsynaptic membrane here under the nerve ending, which show all these receptor molecules and look like they are some pentameric organization. And indeed, these receptors are pentamers. They are made up of several subunits of different kinds. And we were able to purify subunits themselves and to identify the first 20 amino acid sequence from the alpha subunit. And then others colleagues and friends were able to, after me and others, to get the full sequence of the different subunits. And these three groups, and mostly that of NUMA, Heinemann and ourselves, were able to give the full sequence of the receptor and propose a two-dimensional hypothetical model where we propose the occurrence of an N-terminal domain, which would be extrasymptotic and carry the acetyl site, and some 4 transmembrane domains, one of them, of course, lining the ion channel. So this was a highly hypothetical scheme, and of course, the question came immediately: is a nicotinic receptor and allosteric protein? And through biochemistry, affinity labeling, genetics, and so on, we and others were able to identify the amino acids from the acetylcholine binding site at the boundary between subunits, as you can see here, but present in the synaptic domain I just illustrated before, and the transmembrane domain of the ion channel, which was the M2 segment, and show that using either a blocker of the channel, like chlorpromazine, which labeled these hydrophobic amino acids within the channel. I shall come back on that later on. And through also some patch clamp and side-directed experiment from the group of Saxman, show that these outside and outer and inner rings also contribute to the cell. So there is a beautiful picture where our initial results are framed by the amino acids of NUMA and Saxman. So this clearly shows that the acetylcholine site and the ion channel are far distant at the level of the nicotinic receptor protein. This is a higher resolution image by Nigel and Nguyen, and you can see that the distance, which was measured by Palmer Taylor and colleague, is about 40 angstrom between acetylcholine site and 60 angstrom from the acetylcholine binding site and the ion channel. So this is a bona fide allosteric protein, and the relationship between acetylcholine as a regulatory ligand and the ion channel where the ion goes through the membrane is and have to be an allosteric interaction. So to further investigate this transition, which account for the opening of the ion channel by acetylcholine, called activation reaction, we try to purify for years and to crystallize the receptor from the electric organ. The purification was okay. We couldn't have huge amounts, grams of receptor, but the crystallization never worked until the group of Tasnim published a paper in 2005 that there exists in bacteria some sequences which resemble very much, as you can see here, the organization of the nicotinic receptor from muscle and brain. So I immediately realized that this was an important discovery for us. And I asked Jean-Pierre Changeux to try to identify and purify the receptor, one of these bacteria here, Globacter violaceus. And very interestingly, it happened that this molecule was a ligand-gated ion channel, and pH was causing the opening I'm sorry, of the channel. It's a proton-gated ion channel, and the X-ray structure at 3 angstrom resolution was obtained, and you can recognize here the extracellular domain with beta-plated sheet, the transmembrane domain with alpha helixes, and the channel plugged here by detergent. So the interesting outcome of this study was, of course, for us to have the atomic structure of ligand-gated ion channel, again, that of Glick, Dutzler, and others, mainly Guo and Ryan Hibbs, have been able to further crystallize the receptor for glutamate in worms, from GABA in humans, glycine, serotonin, acetylcholine, nicotine from human, and these other subspecies of receptor, the alpha-7 receptor and the muscle receptor, finally, from torpedo at the atomic resolution. And this is, for us, an extremely important conclusion, and you can see that the tertiary and quaternary structure of these different receptors, despite a few differences in sequence. Look at this one, the analogy between serotonin and Glick is amazing. And therefore, we have to say that we have in our brains receptors which are highly similar to those of bacteria and impose their constraints to all the cognitive function we have in our brain. So sometimes people are surprised by the idea that the human brain is so slow. Well, this is due to these allosteric transition taking place through neural networks, and which are themselves under the constraint of what they were in bacteria. Of course, there have been some evolution, but still the range of time scale is the same. So this is the highest resolution we have of the nicotinic receptor, and we have the actual binding site, as you can see here, for nicotine, the ion channel, and of course, clearly there is a topographic distance between the two, and this opened the way, of course, to drug design at the level of the atomic structure for the acetylcholine binding site and for the ion channel. Now, throughout this work, we and others have been interested by compounds which we discovered modulate the transition between, let's say, open and closed channel by binding to sites which are not located at the same level of the neurotransmitter site and the ion channel. So we propose that these would be allosteric modulatory sites, like calcium site for the synaptic domain, and other molecule I shall discuss in a moment at the level of the transmembrane domain. So there is indeed a new category of ligand binding to sites which are not the orthosteric site for neurotransmitter and the ion, and which are of particular interest since most often when you do drug design, in particular by steric analogs of the neurotransmitter, you of course interfere with the brain signaling, while there is no structural analogy at all between these allosteric site and the orthosteric site, and therefore less interference with brain signaling. Therefore, there is a new strategy for drug design which appear. Just to illustrate this, with ivermectin and neuronal nicotinic receptor, this was done with Daniel Bertrand, and we discovered that ivermectin, which is a compound well known from the work of Muir and Campbell as anti-helmintic, which dramatically increase the response of the nicotinic receptor, and mutation in the transmembrane domain actually block the effect of ivermectin on the response, and suggesting that in fact the site is in the transmembrane domain, and here you recognize the transmembrane domain of the glutamate receptor from C. elegans, and you see here beautifully in yellow the molecule of ivermectin, which is a switch in between alpha helices, or sometimes even within alpha helices, but most of all here between alpha helices, and therefore are able to have such a dramatic effect on the receptor through this new category of site. And this is, of course, an important discovery, and is not only the ivermectin site, but also sites for compounds like etomidate, PNU, and even monoclonal antibodies in the case of the nicotinic receptor, and in the case of the glycine receptor, even more than that. One can define 6 categories of allosteric site for tetrahydrocannabinol, picrotoxin, pregnenolone, even ivermectin, and also allosteric site in the synaptic domain. So this is really, I am not emphasizing things too much, a new allosteric pharmacology. Now, of course, if you have this population of different sites on the molecule, you need some conformational change to link them. That was the postulate of the two-state model initially. And we were very early on interested in the identification of this conformational change. First, using fluorescent agonists and using a stop flow. The work was done by Thierry Edman in his thesis work in my lab. That was in the 80s, quite a while ago. And here is the model that we proposed for accounting for the rather complex conformational change, which were monitored through this fluorescent binding. Of course, it's the fluorescence binding of the ligand, which was followed. The idea was there is indeed a two-state transition between closed and open channel with L. Now, the very interesting thing is discovered by Michael Jackson is that this state can open without binding acetylcholine. So this is evident, of course, supporting the shift of pre-existing population of state. Now, there was also a state known for a long time, which is referred to as desensitized state, which is a closed state, slow access state, high affinity state compared to the from the active state, which was already identified by Katz and Teslev many years ago, and that here we can capture through these conformational change. And again, using this technique, one can also show that the desensitized state can be present in the absence of any ligand, and therefore be spontaneously present in the membrane. Now, this opened the way, paved the way to further studies on the conformation transition. And the first study we were doing with Antoine Tally and, of course, Pierre Changeux, Marc Delarue, was to make an in silico model of the nicotinic receptor. This was in 2005, before the final structure of the receptor was actually known by X-ray crystallography. And we developed a method of normal mode analysis to represent the interaction dynamics between atoms as springs. And in the first mode, we identified two discrete states, one closed, you can see here, and one open. And we proposed that this was some kind of a representation of the allosteric transition from the resting to the active conformation, and taking place through some kind of tilt of the subunit and twist, so that there was some kind of link between the synaptic domain and the transmembrane domain. And that was, if I may say, the first kind of proposal which could be made for this allosteric transition. Now, this has been further investigated both by molecular dynamics, by Marco Cecchini, and I wish to mention the name of Martin Karplus, who was co-signed this early paper on the molecular dynamics of the nicotinic receptor, and Marco Cecchini, with whom I still working together. And in parallel, there was the X-ray structure of the closed and open channel state. And you can see that, again, we have a twist which is taking place, and some kind of bloom of the quaternary organization. And therefore, this was some kind of image of the, sorry, I cannot. This led us to propose that there is some kind of two-state switch, which are now being resolved at the atomic resolution, and which is, of course, constrained the propagation of signals through the brain up to cognition, as I already mentioned before. So this transition has been further investigated here by the group of Curie for the muscle nicotinic receptor. And this is a proposal we have made with Marco Cecchini and Pierre-Jean Changeux recently in the annual review of biochemistry for the motion of twist and bloom that one can find from the within the synaptic domain and the link with the transmembrane domain, as you can see here with these arrows. So this is again a representation of the conformation change, which again link together the different binding sites present on the receptor, and we further investigated this transition by this was done by Pierre-Jean Changeux, recently published in Nature, by comparing the structural transition with the channel opening, and the transition being followed by fluorescence, and the channel opening by patch clamp. And you can see that there is not a coincidence in time between the opening of the channel and the fluorescence reaction. And this let us propose that, in fact, there is, you can see here, here is the opening of the channel taking place, but the conformation change followed by fluorescence start before, and therefore there is, as we initially suggested in this very first model, some kind of propagation of the conformation change from the synaptic domain to the transmembrane domain and reverse. And this is an illustration where we are to understand the conformation change taking place within these nicotinic receptor. And as I said, from the work of Curie Edman in 1981, there are more states. I mentioned the desensitized state. We have now the atomic structure of the resting and desensitized conformation. And of course, we can further document this model 40 years, if I may say, after the initial proposal. And I would like to say that one of the main characteristics of these regulatory enzymes is their conformational plasticity. The word plasticity, which I think was introduced by Martin Karpus, is a very important signature of the regulatory proteins and the allosteric transition. Now, I may go even further toward human disease by showing the result of an interesting experiment we did together with Daniel Bertrand quite a while ago, actually. And at that time, we were concerned by these sites, which I mentioned were labeled by chlorpromazine in M2. I mentioned that there was hydrophobic amino acids here, are the two. So we thought naively that perhaps if we were going to mutate these amino acids, we could alter the properties of the ion channel. And the paradoxical finding we made, Daniel Bertrand, when he made the recording, said to me, I am lost. I don't understand what's happening, is that the affinity of the receptor increased by orders of magnitude. You can see here, we go from 100 to 1. And this is the dose response curve of the wild type. This is the dose response curve of the so-called constitutive mutants of the receptor. Of course, the idea came immediately that we were stabilizing the active state or an open state of the desensitized state, which is again in agreement with the idea that we have pre-existing states we can shifted by the ligand in the activation process, but which can also be shifted by mutating here the amino acid of the ion channel. So just to illustrate that this is some kind of mutation of importance for neurology, since it was discovered by Berkowitz after our work that some constitutive mutation in the alpha 4 subunit cause autonomic dominant nocturnal frontaloepilepsy. You can see the child here waking up and having convulsions. And again, this is exactly at the level of some of these amino acids I just mentioned before. So, sorry for that. And we were, of course, concerned by the more general position of these amino acids. And it happened that a lot of mutations were obtained by the group of Engel and Sein at Mayo Clinic for myasthenic mutation, which cause gain or loss of function in the muscle nicotinic receptor. And in green, you saw those which give a gain of function, and in red those which give a loss of function. And very interesting, if you put together the work on the frontal lobe epilepsy and on myasthenia, you find that if you study the position of these amino acids which give the constitutive mutation, they are at the boundary between subunits. So this is, of course, interesting finding, which was done with the late Martin Karplus, for whom I had not only a lot of respect, but a lot of affection. So this is, of course, important to understand these mutation giving altered property of the receptor for precision medicine, because if you have a loss or a loss or a gain of function, of course, the pharmacology which is exactly the opposite for the same target receptor, and you could kill somebody, for instance, by giving curare for a loss of function, but on the unsafe people are having a gain of function. So just to say that one can further analyze the pharmacology along these lines for receptor and allosteric modulators, which could modify the response of the receptor, like ivermectin. I mentioned that already. Here is ivermectin, and it gives a potentiation of, and it was discovered by several groups that, in fact, other type of allosteric modulator potentiate the response, like the type 1, but also alter the sensitization, which I just mentioned before. So to make a long story short, it was shown through recent work with Ryan Heaps, which has been published in Cell, that type 1 and type 2 positive allosteric modulators affect differently the opening of the channel, and that the PAM2 would in fact stabilize and open desensitized state, which is of course in agreement with this four-state model which I illustrated before. So we have now a situation where you can study the pharmacology at the atomic level, at the level of the binding site, at the actually the level of the conformation state, and thus can predict agonists from antagonists and desensitizers. And this is, in fact, a way to have a conformation-specific drug design, which is, of course, a new type of pharmacology, a revolutionary type of pharmacology, if I may say. So this is true for other receptors. I will go quickly to close this lecture. First of all, I mentioned from genome to mind, and here we are at the level of transcription factors, and one can find allosteric modulators of transcription factors which are heterodimers, and some of these compounds act, in fact, as efficient drugs against glioma cell growth, which has been found by Gregory and Zigelny, and using the fluorescence cross-correlation spectroscopy with the group of Lars Tyrenius and Igor Zigelny, one can show that these compounds indeed bind to a specific site at the interface between two subunits. So other receptors like the GPCRs, like the muscarinic receptor, show positive allosteric modulators sites, which are different from the orthosteric sites. Here is the detail. This is from the work of Kobilka and collaborators. And very interestingly, recently, there have been the discovery of, by the group of Engers and Temple of a positive allosteric modulator of the muscarinic M4 receptor, which is a very efficient anti-psychotic drugs. This is a compound which could be more efficient than the orthosteric agonist, xanomeline, which is already currently accepted for FDA, and open antipsychotic agent from a new field, cholinergic field, while in the past, they were mostly known as modifying the dopaminergic system. So this is, in fact, a new orientation which is taking place for the pharmacology of antipsychotic drugs. The same group of Kobilka also discovered an allosteric modulatory site on the opioid receptor, which is, of course, very important to minimize withdrawal sign. And here, just to say that in cystic fibrosis 3 therapy, there are 3 drugs, 2 which are allosteric modulators, which modify the folding of the protein, and ivacaftor, which is acting here on the trans membrane domain, again, which is an allosteric modulator of the CFTR protein. So this just illustrates that there are many successes in drug therapy. And here it's not a complete list, but I, together with Arthur Christopoulos, we listed about 91 allosteric modulators which are available as medicine. And in addition, there are many new conformation of specific monoclonal antibodies which are being developed for different kind of receptors. So as you can see, there are drugs which are known to, of course, act like the diazepam, which were discovered very early on, many years ago, for anxiety and insomnia, which is a positive allosteric modulator of the GABA receptor. They are for negative allosteric modulators for the calcium channel in hypertension, some HIV compound and cancer compound, and so on and so forth. So there are indeed a lot of new compounds coming up, and this is a pharmacology which is, of course, very different from the early pharmacology, competitive pharmacology, which was proposed by Sir James Blacks 50 years ago. So we are now in a new situation in the case of drug pharmacology, and the take-home message is first of all, the validity of the concept of allosteric interaction between topographically distinct sites, the two-state model, which is too simple. In fact, there are more than 2 states now, we know. There are this new category of drugs which are referred to as allosteric modulators. The allosteric transition can be studied at atomic resolution, and this opened a new way of drug design, which would be a conformation-specific drug design. Thank you very much.
Thank you very, very much, Professor Jean-Pierre Changeux, for this very extensive overview of the work you carried out during all these years. I don't know if people have any questions in the audience.
Maybe you could change me the screen if I could see the people.
I will try to. prevent you from sharing your screen, but I don't know if I can do that. You have to stop the sharing by yourself, I think.
I stop sharing, okay?
Yes, yes. And then you should be able to see the gallery of participants.
No,
Yes,
okay, thank you.
perfect.
Very good.
Yes, so I will open with one question. I was very surprised by the amount of allosteric regulators that can bind on the acetylcholine receptor. And
Yes.
I was wondering if there is a possibility to predict from, I don't know, molecular modeling, for example, to screen for compounds and find out new modulators. You probably know about drug repurposing, which is a very timely concern at the moment to find new effects of drugs. And I was wondering whether drug repurposing could find an application to identify new allosteric modulators of any kind of proteins.
Yes, 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 yes. You are perfectly right. There is a strategy ongoing there. Sometime using with care the AlphaFold story. Of course, we don't learn anything from AlphaFold about the folding of protein, but we can nevertheless use a very efficient tool to try to predict the conformation. And from the actual X-ray, EM, and AlphaFold story, one can try to investigate sites at the level of the molecule, which could be potential regulatory site. Of course, the AlphaFold does not give any physiology compared to all the work which was done before with allosteric protein, of course. But at least going along what you say, this is a new possibility which is being opened. And I may say that many years ago, Max Perutz, from the X-ray structure of the hemoglobin, tried to predict all the binding sites at the boundary between subunits in the diverse axes of symmetry, developed compounds which were binding to these different kind of sites. That was almost 50 years ago. So I was a student at that time. And unfortunately, the sites he predicted were correct binding sites, but the affinity was not very good. So he was trying to get a compound against single cell anemia, and none of them work. So this is also something we have to keep in mind. And that's where, when you look at binding site without following binding, you may find sites, but which may not be efficient as TOICs. Maybe there are other questions.
Roman?
So fantastic lecture. You have shown many structures which are highly symmetric, and you may mention the word symmetry several times during your talk.
Yes, yes, it is, indeed.
And I have a general question. It's a bit puzzling that there are so many structures found in nature are symmetric, when in many cases, symmetric structure is not the lowest energy structure. So what is your opinion on the role of symmetry in biology?
You know, there was actually a meeting in Stockholm in the 70s, which was very interesting on symmetry, which was organized by Peter Reichhardt. I don't know if you remember him. And that was, I think, a Nobel Symposium. And this debate was between, I would like to say, idealistic philosophical views like Platonist views, if I may say, and more down to earth, more concrete Aristotelian views. One claiming that the symmetry causes the cooperativity, and the other being that simply the fact that they have to assemble a subunit to make an oligomer which mediate cooperative interaction simply is accompanied by symmetry, because once you try to associate a subunit to make an edifice, then there are symmetry properties which are appearing. So I think there was some kind of a debate between Jacques Monod and myself. He was more on the Platonist side, I was more on the Aristotelian down-to-earth side. So I think the debate is open. Just to say it, but it's correct that your point is a very important one. It's an amazing one, and to my opinion, what is important is the regulatory function, which is, usually one subunit is not regulatory. Well, there are some regulatory. I had a big debate with Martin Karplus about that. You can have allosteric protein which are monomers, but in most of the cases, most of the regulatory proteins you have from the genome to the mind, if I may say, are in fact resulting through evolution from the association between subunits. And if during my talk you may have remembered that I insisted on the interface between subunits is simply because of that. And most of the regulatory sites are precisely at the boundary between subunits. So I would not be too much concerned by symmetry. I am, but not too much. I am more concerned by the oligomeric structure. And I think this is something to look at very carefully. And you have not only the case of small globular oligomer, you have the membrane oligomers, but you may have, of course, the filaments and contractile muscle proteins and so on, where you have an extension of allostery to long protein chains. So thank you for asking this question, which is a very relevant one.
Thank you. If I may, I have another question, which will interest some people here as well. You have shown molecular dynamic simulations of huge structures, up to 200,000 atoms.
Yes.
And I know that all these simulations are done on the assumption that these are purely monoisotopic molecules. All carbon atoms are considered to be C12. All hydrogens are hydrogens, no deuterium, no N15, and so on. But if you have 200,000 atoms, half of them are carbons, 100,000, and C13 is 1.1%. So 1,000 atoms in that structure are C13, actually, not to mention other isotopes. And they have different angles. They have a little bit different bond strengths. How will that affect the output of molecular dynamic simulations?
I'm not a specialist. You may ask Martin Karplus and Marcus himself about this issue. But what I want to say is that in biological sciences, of course, we are making models.
Yeah.
And I try to illustrate how we started with a very simple minded kind of model, and then progressively develop model from the X-ray structure and the molecular dynamics and so on and so forth. But they are always models. And I don't believe myself that any kind of model can exhaust or fully describe the reality of biological objects. So we have to be humble as modelers and have to accept that there are deviations, and there are. So it would be wise to follow up what you say and see how far the deviation can have to be considered under the circumstances you mentioned. I think this is a work for the future somewhere.
Okay, thank you very much.
Yes.
Laszlo, you have a question.
Hi. Hi. Can you hear me?
Yes.
All right. So you were at UCLA or which institute in California?
I was no. I was in Berkeley.
Oh, okay. Did you know Judy Kleinman by any chance?
Who?
Judy Kleinman. She was the
No.
head of the doctor school,
Mm-hmm.
Stanford. But anyhow, so did you happen to know Paul Boyer at UCLA? He discovered these ATP synthase nanomotors in mitochondria.
Mm-hmm.
And we worked on the subunit C, which sits on the top of this shaft that rotates. It's a very highly rotating enzyme which transfers protons from the intermembrane space into the matrix. And it's a highly deuterium sensitive enzyme protein that rotates. And I'm just trying to, because Dr. Zubarev, Roman asked the question that I wanted to ask actually about isotopic conformational changes and dynamics of these protein networks, and you answered that question just like that's the future. Now we have a field we developed together with Roman called deuteronomics, which is practically the hydrogen replaced by deuterium and the protein or peptide or enzymatic and tunneling response to this atomic single exchange reaction, simply because they have actually dramatic, in some case, fundamental effects on tunneling and enzyme activities that we are pursuing now. And it clearly connects with your work. And simply my question was just first, I would assume that we have to kind of adjust or set a deuteronomics or a isotopic profile of the protein that we are profiling, and then coming with modulators of conformational and other regulatory changes in the protein networks. And this is, I just wanted to kind of support your theory of what the future will look like, but practically it seems like isotopes will have a major role in this entire proteomics or enzymatics or tunneling reaction scenarios and realms. Thank you so much.
No, I thank you for the comment. And there is still work to be done after 50 years. So, thank you very much. Are there other questions?
Is there any other question? I have maybe one small last, because I would like to go back to the personalized medicine that you mentioned.
Yes.
And the very different effects that you may anticipate depending on the sequence each individual has for a receptor. And you mentioned that sometimes drugs would be predicted to act in a way, but they could have very adverse effects.
Yes.
So how would you proceed in order to avoid medical errors?
But this is, this issue was actually studied by Sign and people I mentioned at Mayo Clinic with the myasthenic mutations, because they have been able to really identify not only the mutation itself, but the phenotype which goes with the mutation. And they actually found that compounds which were antagonists of the receptor which were extremely dangerous with negative mutation, when they was put, were extremely useful for positive allosteric mutation. That was what I mentioned is the work they did. And I think this is something which we may not think about. I was recently at a meeting in Paris on psychiatry, and many people are making statistics for drug action on patients, and they found usually bimodal or very flat kind of statistic and so on. And this is, of course, due to that I discussed with them, to the heterogeneity of the phenotype that you get with a given set of mutations. So this is, raising a question whether one should develop drugs which are based on the genotype, on the type of mutation, or on the phenotype, which is what do you observe actually in the body of the patient. So I think the precision medicine at the gene level is, I would say, some kind of utopia, because it would mean that you not only have to go with your having in your pocket your genome, your sequence, but in addition, you should be able to identify, to ascertain what type of mutation, what is the phenotype of this particular type of mutation. This is theoretically absolutely possible, but practically very, very, very difficult. And in case of mental disease, there is an additional paradox which is not solved, which I am very interested in, is the fact that if you look at autism, autistic mutation, or schizophrenia, this is work of Thomas Bourgeron and a lot of people throughout the world, you find that there are maybe 300 predisposition genes for a common phenotype. You know, the Asperger disease, a la Elon Musk, for instance. No, but that's true. He said,
Yes.
I am not saying things which are not public. I think he himself claimed that he is Asperger. So I think in this kind of mutation is extremely difficult to relate the gene with the phenotype. On the other hand, you find for the psychiatrists a very common phenotype for many different patients with different genotypes. So the issue is whether the design of the drug should not be against what is genotypic, but against what is phenotypic for the brain. So this is really a very difficult dimension, but pharmacology is not easy, that's for sure. But I think it's an extremely important discipline for the future, and in the brain we need many more pharmacological agents, for instance, against Alzheimer's disease, against depression, against schizophrenia, autism, and so on. So there is an urgent need of new compounds.
Thank you very much for your input. So if there are no other, yes, there is one more question from Amir.
Thank you, Professor, for the inspiring presentation. I work with metabolite protein interaction, and I kept thinking, because your discussion, I mean, your presentation was mostly about the receptors. I'm thinking there's the same, like, apply to across the whole proteome, across also the intracellular proteome. Do you think if we study them, we will find more and more allosteric sites? And is this something that I mean, some of these are, of course, evolutionary, but is this also because we study them that we find these kind of, like, especially the positive allosteric modulators? And what is the evolutionary advantage? Or is it, I think you know what I mean.
No, I think your question is about the positive allosteric modulator.
Yes.
Yeah. And their evolutionary significance. Well, there are two possibilities. First of all, they could be regulatory sites for ligand, which we do not know. You know, and if you look at the case of the glycine receptor, I showed where there are 6 categories of, you find among these regulatory ligands THC, tetrahydrocannabinol. So the question is whether it is a natural kind of modulator which is present on this site and having within the membrane some role. Now, in the case of ivermectin, I don't think it is not known whether there is some natural effector which is able to take this place. But just in 2 words, in some case, you have natural ligands which are binding to this site. In other cases, they are purely artificial ones. So I have no preference myself. I think we have to analyze them, and the idea that you may have more regulatory ligands than you anticipate in the brain is something that is very plausible. On the other hand, the pharmacology we know presently about allosteric modulatory site is such that many of these compounds look very artificial. So that's also a reason why many of us claim that the allosteric modulators are more efficient, or I would say better pharmacological agent, because they do not interfere with actual regulatory mechanism. You know, they are completely new to the protein and usually highly specific to even a particular subunit in an oligomeric molecule. So there is still a debate there. In any case, they are very useful. That's for sure for pharmacologists. Are there other questions?
Yes, Max.
Hi. Thanks for the previous questions because it triggered my question. And so do you think, what's your vision on the possibility of having allosteric modulation also, not just by drugs, but by proteins that are in the same complex that we you know, or near complexes that we can trigger with drugs? So relating to Muriel's question, do you think we could have, you know, when we screen or when we try to do some selection, for example, of repurposing, drug repurposing, when should we have some databases to kind of guess maybe some modulation by cell proteins to our target for allosteric modulation of that function? So would that work in your opinion?
Yes,
should that be included? Because currently is not.
I think you may have allosteric interaction simply by protein association.
Yeah, that's what I mean. Yeah.
To the regulatory oligomer I just mentioned. And if you remember the first slide I showed, which was on the life and so on, from human genome to mind, which is very just a little bit a joke to some extent. But in the left picture, I show the transcription factors. I showed a slide of a model of how transcription factors are acting on the polymerase. And the issue is apparently now that the transcription factors are associated between themselves to make some kind of regulatory multi-protein complex, which is regulating the transcription either on DNA or DNA RNA. mostly DNA, RNA transcription. So this is a future. I think one should look in the future to this kind of interaction. And the possibility of allosteric interaction in relation with protein-protein interactions, in particular, different categories of protein, different particular proteins among this given category could be extremely useful things to follow. And I took the example of the transcription factors, but there are many more systems which are, if I may call it, supra-macromolecular kind of organization, where you anticipate to have allosteric effects.
Thank you.
Thank you very much. More questions? No, nobody raises their hands. So I think it's time to thank you one last time, Professor Changeux, for being with us tonight by video conference and for sharing all the knowledge that you could deliver tonight. Thank you very much.
I am very pleased, and I thank you very much for your patience.
Thank you.
And for your consideration. Thank you so much. Thank you very much.
Thank you very much.
Have a good meeting.
Thank you. You feel free to join if you want to know more about the future talks.
Okay. Thank
All
you.
right.
Bye-bye.
Bye. It's time to close the session for tonight, and we can meet tomorrow again at 9. Bye.
Bye-bye.